I'm Germino. I'm a grad student at UCSF in the biomedical sciences program, and I'm jointly co-mentored by doctors Jay Gardner and Sil Centranos. I'm mostly interested in studying the mechanisms responsible for regulating the immune system. So under normal conditions, it's very important the immune system is equally balanced between stimulation and repression, as if this balance is disrupted and the immune system becomes overstimulated, you can develop an autoimmune disease, or if the immune system becomes overly repressed, you can develop cancers and escape tumors. One of the key effector cells that are central to these immune processes is a group of cells called T cells, which are able to recognize peptides associated with pathogens or tumor cells, and then induce the death of those pathogenic or tumor cells through a number of different mechanisms, mostly involving the secretion of immune cytokines like interferon gamma and granzymes. However, it's very important that T cells are educated to discriminate between self and non-self cells, as if a T cell is able to recognize and induce the death of self tissues, you can develop a very severe and serious autoimmune disorder. So this process of negative selection or T cell self-education occurs in the thymus, where developing T cells interact with a very special population of epithelial cells called MTECs, who are able to express and present a very diverse repertoire of self-antigen peptides. And then any T cells that are able to recognize these self-antigen peptides are removed via clonal deletion or inactivation, so that the only T cells that complete their development in the thymus and escape into the periphery as mature effector T cells are those cells that are able to recognize pathogenic peptides, but not self-antigen peptides. And this process of expression and presentation of self-antigen peptides in MTECs is under the control of a very unique transcription factor called AIR. But interestingly, recent studies have suggested that other transcription factors, such as the brain transcription factor FESF2, are also expressed in MTECs and may contribute to a smaller extent to the expression and presentation of these self-antigen peptides. So my research goals mostly focus on understanding FESF2's role in MTECs, in particular understanding how it may influence MTEC development and differentiation into terminal epithelial cells of the thymus that mimic peripheral epithelial cells. I'm also interested in exploring alongside this the self-antigen repertoire that's induced by FESF2, as well as identifying other transcription factors that, alongside FESF2 and AIR, help to regulate self-representation in the thymus. And finally, because FESF2 has a known role in the development of neurons in the central nervous system, I'm interested in looking to see whether other neurodevelopment gene regulatory networks are present and functioning in the thymus, both for self-antigen expression and MTEC development. And with that, I'd like to thank ARCS for their support, as it's enabled me to really focus on understanding better these mechanisms of the immune system and hopefully being able to identify how these mechanisms may become dysfunctional in autoimmune disorders.